Good afternoon, my name is Sofia Zuluaga and today I am here to present the research project developed in Energy and Memory team titled Device for Optogenetic Activation of Neurons in Drosophila melanogaster. This laboratory study the links between energy metabolism and memory. Memory neurons are known to receive glucose from glial cells, which are found around them to support. When neurons receive glucose, they can form long, medium, and short-term memory. However, when the passage of glucose is interrupted, memory formation is lost. It is also known that acetylcholine is a neurotransmitter responsible for activating glial cells, although it is not known where it is come from. The energy and memory team hypothesized that it is the same neuron that activates glial cells by giving them acetylcholine. They also hypothesized that by regulating the amount of this neurotransmitter, long-term memory can be blocked without affecting medium and short-term memory. This is why the laboratory is asking the question of how to control the supply of acetylcholine to manage only long-term memory. First, a way must be found to control a specific neuron without affecting memory formation. That is why they were thinking about using optogenetics to with light activate a motor neuron. Why a motor neuron? Because they can give a feedback just after the illumination. The objective of this project is to build a device to activate neurons using optogenetic compatible with behavior conditioning from the laboratory without disturbing memory formation. For this purpose, we use Drosophila line that expresses CS crimson and Venus in a specific subset on a motor neuron. So, when the flies are fed with retinal and exposed to intensive red light, they begin to walk backward. The retinal is sensible to the light. That is why a device must be constructed to keep the flies in the dark place while feeding. Also, a second device must be constructed to expose the flies to the light. We must check first the expression of the pattern of the CN crimson and Venus. On the left side, you can see from the literature what the gene expression should look like. So, first, we put the flies in PFA overnight at 80 degrees Celsius. Then we made the dissection and mountain. 24 hours later, we imagine in the confocal microscope. Here you can see different images that I took that show the expression of the gene. And should be noted that I did not amplify the signal without any antibody. Second, we have to test the functionality of the device. For this end, the transmembrane protein is activated with red light. This induces the entry of calcium to the motor neuron, then the detection of moonwalk behavior is done. So the entire protocol is put the flies in retinol in a concentration of 1 millimole in the food media, then let them for 3 days at 18 degrees Celsius. After that, we do the memory conditioning that is basically exposed to the flies to two other, one related to electroshock and to the other one that without electroshock. And just after that, we do the illumination protocol for 5 minutes. Then we have to put the flies again to the dark place in the dark box for 24 hours at 18 degrees Celsius. And after that, we do the memory test. So when we put the flies in retinol and we just expose to the red light, we can see like the fly is going backwards. And then if we put a track in the butt of the fly, we can see the trajectory of the fly and corroborate that it is going backwards. In this second video, it is the same experiment and you can see the behavior. Here, the same flies with our retinal and LED. They have a normal behavior. In the final part, we did the memory score and we conclude that even the retinal or just the illumination of the combination of both of them do not affect the memory scores. Finally, I would like to extend my sincere thanks to Jean-Claude, who helped me with build the device, Samuel Hidalgo, for letting me the controlling, helping me with all the electronic parts of the project, for the members of the laboratory for helping me with the memory tests and letting me the different laboratory equipment, and finally to Alice Pavlovsky for letting me be part of the team and for the continued support throughout this project.